Hi Mindsetters and welcome. Grade 11s, I'm hoping you're working very hard. Today we look at photosynthesis. Let's look at that word before we even look at the lesson itself. Let's look at this word photosynthesis. If we break it up, we get the word photo and synthesis. Synthesis, to make. Photo, light. So make something using light. And that's what this lesson is all about. We're going to look at how we can make something. We're not going to let out what that something is until the lesson unfolds using light energy. What do we have to focus on in this lesson? What are the key points that we're looking at? First of all, we're looking at the process of photosynthesis using words and symbols. So we're going to explain the process of photosynthesis with words, and then we're going to explain it with using symbols. In other words, in a form of a formula. We also want to know, why does this photosynthesis take place? In other words, why is it important to life? Importance of photosynthesis. We then move on to look at the effects of variable amounts of light, carbon dioxide, and temperature on the rate of photosynthesis. In other words, what do these things do, these variables? What are these variables again? Light, carbon dioxide, and temperature. And we're looking at their effect on the rate of photosynthesis. In other words, do they speed up the rate of photosynthesis? Do they slow it down? Do they have no effect? This is what we want to look at here. We also want to look at the role of carbon dioxide enrichment. In other words, if we add more carbon dioxide to a system, how does that help in a greenhouse system? Not only carbon dioxide enrichment, but also optimum light. In other words, the most perfect light. Not too little, not too much, just right. We want to look at that in terms of light and optimum temperature. So in other words, if we put all this in a curly bracket, we are looking at ideal conditions or favorable conditions for photosynthesis to take place. If we do, we create this utopia, we create this ideal environment. What will happen in this greenhouse? How does it work for us? Lastly, we also want to look at the role of ATP as an important energy carrier in the cell. Remember, ATP means A with P, P, and another P. This last P, the third P, adenosine triphosphate, ATP. Fortunately for you guys, you don't even have to say adenosine triphosphate. You just say ATP is good enough. But the trick here is that this last phosphate is associated with energy. And we're going to look at how ATP forms and how this energy is used in the process of photosynthesis. And later in other lessons, you will look at how this ATP is used in respiration. So we have now gone over what we're supposed to do. Let's not waste time. Let's get right into this thing. So now let's explain what's happening. First of all, the question asks, what is photosynthesis? And in words, we said we're going to explain in words. Photosynthesis is the process whereby green plants use the radiant energy of the sun and the carbon dioxide to manufacture their own food. Remember when we explained the word photosynthesis just now? Photo, using the light to synthesize something. What's that something? Food. So not only the sunlight is used but during this process, but also we use carbon dioxide. So we're taking carbon dioxide, which is a raw form. We're taking sunlight, which is another ingredient in its raw form. But we're converting those things. We are involving them in a series of reactions where some transformation will take place. And this transformation will result in the production of food, which is usable to plant itself, the plant that's making it, and to the animal that eats that plant. And in that way, this food becomes available in the ecosystem. Wow, amazing, amazing and more amazing. The more you do life science, guys, the more you understand the wonders of nature and the creation that is in front of us. And all these things we are only understanding now. So let's look at it now in terms of a diagram. Let's look at a diagram first. What we are saying in our words is that light energy is coming from somewhere into this plant, and we're talking about green plants. Notice why green? Because they have chlorophyll, and chlorophyll can trap this radiant energy of the sun. Carbon dioxide comes into this plant as well, and together they form food, in this case forming glucose. Notice here on top, oxygen is released. So oxygen is another product, but this product is not used in the photosynthesis. It's not food. This oxygen is released back into the environment. Also, another requirement would be water. So if I had to ask you, what are the requirements of photosynthesis? You would have to tell me, 1, chlorophyll. The plant has to be green. 2, radiant energy or sunlight. 3, carbon dioxide. And 4, water. These are the 4 requirements of photosynthesis. So what then are the products of photosynthesis? You got it? Obviously, the food that is formed, glucose in this case, this glucose can become starch, it can become proteins, it can become fats, can be converted to various food sources as such. And another product is oxygen. But the oxygen is a byproduct. It's given off, its release. Now, our first bullet on the top, when we started, we said we want to explain in words photosynthesis. Now, I have just used the diagram to explain the process in words, and we also said in symbols. Here's the words here again. If we put it in a scientific formula, carbon dioxide plus light plus water gives us sugar and oxygen. But all this must happen in a chlorophyll molecule. Scientifically, that's just words, so it's not good enough. A scientist wants a formula. He wants a formula to say what is happening there. So we start by balancing an equation. Carbon dioxide plus water gives us C6H12O6 plus 6O2. You'll notice that the way this thing is done is that the equation is balanced. So these are the symbols. So we have not just carbon dioxide, but 6 carbon dioxide plus 6 water gives us glucose, which is C6H12O6, and oxygen, which is the byproduct. And obviously, this is under the control of enzymes, and we need sunlight to come in there, and we also need chlorophyll there. It's not part of the formula, but that is the general formula for photosynthesis. So we have answered the first bullet in that, explaining photosynthesis in words and in symbols. There, another diagram showing you the same thing in a little bit more graphic form. We don't have to go through that because we've done it already. Photosynthesis takes place where? Mainly in the leaf. Why? Because they contain chloroplast. Just don't be fooled though. Notice we're saying that it takes place mainly in the leaf. We didn't say only in the leaf. We said mainly in the leaf. Because photosynthesis can also take place in the stem if the stem has chlorophyll. Remember that. Please keep that in mind. Okay, and what are chloroplasts? They are tiny, not objects, but they are organelles. If you did your grade 10 work, you'll remember that chloroplasts are organelles that are found inside green plants. And they contain this important pigment called chlorophyll. Chlorophyll gives the leaf its green color. And that's not all it does. It absorbs sunlight. It traps radiant energy. And inside this chlorophyll or chloroplast molecule, all these things will take place. We move on. The organ of photosynthesis, therefore, would be the leaf. And here we have a cross-section through the leaf showing you the upper epidermis, the mesophyll, and the lower epidermis. Just above the epidermis, you see this tiny layer here will be the waxy cuticle. Your grade 10 work will teach you that the waxy cuticle is there to protect the plant from dehydration, in other words, from losing too much of water. Just below that is the upper epidermis, and below that is the mesophyll. The mesophyll in turn is divided into two parts. These elongated cells here, these ones here, called the palisade mesophyll, and below that the spongy mesophyll. You notice these little dottikis here? These dots represent chloroplasts. That means all of these cells have chloroplasts. And if they have chloroplasts, they have, you got it, chlorophyll. And if they have chlorophyll, they can, that's it, trap radiant energy from the sun. Okay? Now look at the bottom layer. It's slightly different from the top layer. We also have a waxy cuticle there. This is called the lower epidermis. And notice we have some specialized cells there and there. These cells are called guard cells, and the space in between is called a stoma. Now, one of the principles of marking is, if a child makes a spelling mistake, accept the spelling mistake if it does not mean something else in life science. For example, this is a perfect example here. Stoma is the opening in the lower epidermis of the leaf, and stroma is the ground substance found in some organelles. For example, stroma is found as a liquid in mitochondria. So notice, you can say it's a spelling mistake, but because something else in life science has this, then it's marked incorrect. I'm going to take that away so we do not confuse each other. So just to explain that you need to know the difference, and you need to just watch your spelling. It's very important. So basically that is the leaf structure. This area here is the area of highest photosynthesis. Can you guess why? You can't? I'll tell you. It's the first layer beneath the epidermis where the sun's rays will hit. So it's the closest to where the sun's rays hit, and therefore they are in the best position to convert radiant energy into potential chemical energy, which is food, glucose. Also notice the shape of these cells. These cells are elongated, very longish, and they are tightly packed like columns. How does this help? There's more of them. If there's more of them, then there will be more chloroplasts. And if there's more chloroplasts, more photosynthesis. Therefore, this area is the most suited. So this area here, the spongy mesophyll, has many air spaces, as you can see. There, there, there. So this is the spongy mesophyll there. Spongy mesophyll. All of this area here that I have bracketed here. And all the air spaces, as you can see. Lots of air spaces. And air spaces allow for gaseous exchange. It allows for the gases to be exchanged. And remember, here at the bottom you have the stoma. The stoma allows gaseous exchange as well. In other words, something can come in, something can go out there. The carbon dioxide that we need for photosynthesis is going to go in that way. And from there, it will diffuse to all cells in the area. And when we're moving in these areas, obviously, it can go through the air spaces to other cells in neighboring cells in any direction that we can think about. So we said already, photosynthesis occurs mainly in the palisade mesophyll, which contains chloroplasts. I would say contains most chloroplasts. And chloroplasts, by the way, are organelles which contain chlorophyll. We've explained that earlier already. Good. So we're still busy with it. And now we're going, notice what we've done. We've spoken about the process of photosynthesis. Then we looked at the organ where it happens. We looked at the tissues within that organ, the leaf. Now we mentioned chloroplast all the time. Now we want to go inside that chloroplast. So notice what we've done. We've started on an overview. We've gone to the organ, From the organ, what's lower than an organ would be tissues that form the organ. And inside these tissues are cells, which we have spoken about. And in the cell are organelles. So we're going into the microscopic structure of the plant. And we want to say, because if you look at the leaf, can you see chloroplast with your naked eye? No, you can't. But you can see the leaf. You can't even see palisade mesophyll. You can see the effects because the leaf is green, but you cannot see the actual cells because they are microscopic. So now we've gone even further into the cell. And we're looking at the chloroplast. The chloroplast is made up of a double membrane, as you can see, an outer membrane, zapaduda, and an inner membrane on the inside of that. There we're having that one there going. I'm just following it down so you can see. So you got an outer membrane and you got an inner membrane. There's the inner membrane, outer membrane. In addition to that, inside this chloroplast, we have ground substance. There. This ground substance is called stroma. Just like in the mitochondria, the ground substance is called stroma. Notice stoma is the opening. Stroma is the liquid that's found inside the chloroplast. And as we can see, guys, there's a lot of plate-like discs that are found inside the chloroplast. Many of them. Each bundle there is called a granum, as is labeled there. And each disc, the single disc there, 1, 2, 3, those are called thylakoids. So the bundle, grana, granum, one granum, many grana, and the individual plate-like discs that make up the grana are known as thylakoids. And inside this thylakoid, inside each of these thylakoids, we will find the chlorophyll pigment. That is where the chlorophyll is found. So can you understand the system that we're finding here? There's so many of them. What does that do? The more there are, what is it? Yeah? The greater the surface area. The greater the surface area, the more chlorophyll can be attached. The more chlorophyll that can be attached, the greater the rate of photosynthesis. So can you see, we are talking about how the plant is suited for photosynthesis. Okay, this diagram is a micrograph of the diagram. Remember, this is a drawing. This is a drawing. This is a micrograph taken. And you can see clearly here the membrane. You can't see both the membranes, but you can see at least the outer membrane there. You can see the grana there. You can see the stroma, the fluid part. And obviously each line there is the thylakoid. Firstly, the membrane is permeable to water and carbon dioxide. This means that this membrane allows water and carbon dioxide to go through it. What's so good about that, you ask? Why does it happen? Easy, guys. Because the organelle needs water and carbon dioxide because they are requirements for photosynthesis. And if they are requirements, they need to go through. If we don't allow the requirements to come in, the process won't take place. Secondly, the stroma, that liquid part that we spoke about, contains enzymes for photosynthesis. So they help the cycle to continue, the dark phase. We'll talk about that later. The stroma also has ribosomes, which manufacture proteins, and if they manufacture proteins, they manufacture enzymes, because all enzymes are protein. You learned this in grade 10. The lamella or thylakoids, another word for thylakoids is lamella, contain chlorophyll to trap the radiant energy. And we can go further that there are many of them. And if there are many, it increases the surface area. Okay. Lastly, the grana consists of stacks of thylakoids to increase. All right, I've already done that there. It's the same story there. Same thing. Increases the surface area for sunlight, absorption of sunlight. Now we're going into the process, guys. We've looked at the organelles. We looked at the overview. Now we want to know What is this photosynthesis that we're hyping about? What happens? Okay, so let's look at that. First of all, two reactions make it up. We have what we call the light reaction or light dependent reaction, or we can call it the light phase. So that's the first phase that we talk about. That means this phase is dependent on light. Light is necessary for this phase of photosynthesis. And the second part is the dark phase, or we call it the Kelvin cycle, because it was found first by Melvin Kelvin, a scientist, and or we can also call it the light independent reaction. Notice the words, you must be careful here. Light dependent reaction, light independent. Light dependent is depending on light, light is necessary. Light independent, no light necessary. So what happens here? In this phase, energy is produced from solar power, photons, or radiant energy of the sun, in the form of ATP and NADPH. So these are the products of the light phase, ATP and NADPH. We're going to look at the light phase in more detail. This is an overview. Okay? And the dark phase uses this energy. Which energy? ATP and NADPH from the light phase, and it uses carbon dioxide as well, plus carbon dioxide, to give us food in the form of glucose and other organic compounds. And in this dark phase, oxygen is released as a byproduct. I'm hoping you're with me, guys. Interesting stuff as we're going along. Now let's look at each of these phases in more detail. The first phase is the light phase again. Where does it take place? The first question you have to answer is, where does it take place? It occurs in the thylakoid membranes, inside the thylakoids. We also hear in textbooks or read in textbooks, the light phase in the thylakoids. Why? Because the light phase takes place there. What happens here? First of all, there are two possible routes. When the radiant energy is trapped, it can cause one reaction in one way, or it can cause another reaction in the other way. Obviously, both these things happen inside. The first one is called cyclic electron flow. Cyclic. The word cyclic tells you there's a cycle. Okay? What happens here is that the energy that comes from the sun is used to form ATP from ADP plus P. Remember? A plus P plus P. That's ADP. Plus P gives you ATP. So in other words, the energy is used to combine this phosphate here to this, this phosphate here to this molecule here. So we have ATP. This is what it's all about. By the way, ATP is the energy carrier. Why? Because this last phosphate is associated with energy. When the cell needs the energy, then this ATP is reconverted from ATP to ADP. So we use that phosphate for energy later. So now what we are doing, this is what's happening there. Eventually, this electron that helped to make this ATP from ADP, it returns to the original chlorophyll molecule. Therefore, we say it is a cyclic. You don't have to know that part, but it's nice to understand why we call it the cyclic. For your syllabus, it's not necessary. But when you go to varsity, someone's going to ask you, and you're going to have to know that as well. Non-cyclic electron flow. Non-cyclic. So that means here, there's no proper cycle that's taking place. Here, the same energy is used to split water into high energy hydrogen atoms and oxygen. In other words, H2O is split into H2 plus O. All right. This oxygen is going to be released. The hydrogen atoms combine with coenzymes which take them into the stroma, into the dark phase. Okay, so we're getting here NADPH. There's the H that we're talking about. That's the story that's going to happen here into the dark phase. Now what happens in the dark phase? Where does this dark phase take place? It takes place in the stroma, in that liquid part that we spoke about earlier. What happens here? Carbon dioxide from the atmosphere that diffused in through the stoma, the opening, and then went into the mesophyll cells, and from the mesophyll cells into finally the stroma of the chloroplast organelle. Carbon dioxide combines with high energy hydrogen atoms in a series of reactions. That means there's many reactions that take place to form energy rich carbohydrates. So there's a whole lot of transformations that take place with carbon dioxide and hydrogen. Where does the hydrogen come from? It comes from the light phase, NADPH. So the hydrogen from there comes. Hydrogen, by the way, is also associated with energy. When we talk of the atom bomb, we talk of the hydrogen bomb. The main component of the atom bomb was hydrogen. So hydrogen is also associated with energy. The high energy hydrogen and ATP from the light phase provide the energy there. All reactions are controlled by enzymes. So what's the product of the dark phase then? The product of the dark phase is glucose, starch, and any other food. Now, let's look at how our knowledge on photosynthesis can help us in our lives. So we're looking at maximizing growth. Farmers, you see, they can use their knowledge of these limiting factors of photosynthesis to increase crop growth in greenhouses. For example, they have the greenhouse, and inside the greenhouse they have crops that are growing. They can use what they know about photosynthesis, in other words, the requirements of photosynthesis, to do this. First of all, they may use artificial light so that photosynthesis can continue beyond the daylight hours. So in other words, the plant is fooled into thinking it's daylight all the time. Instead of when the sun goes down, now no more photosynthesis. And you take countries like United Kingdom, etc. In winter, their days are very short. We in South Africa, I mean, our shorter days end at about 5:30. But in the United Kingdom and other areas where the winter, they have very short days. 3:30, 4 o'clock, the sun can set. So in those days, I mean, photosynthesis is very limited. And think about the United Kingdom and think about how much sun do they have in the daytime anyway. So these farmers now, what they do, in the greenhouse, they provide artificial light. And the plant uses that artificial light to maximize photosynthesis. So in a higher than normal light intensity, they can use shade netting to reduce light. Let's say now understand what we're saying. There's two things happening here. That's one thing. And above that is another thing. If they want the plant to produce more food during the night, then they provide artificial light for it. And in other areas where the light is so bright that is no good for the plant, then they can use shade netting to reduce the light. So they can use either or strategy depending on the circumstances. The use of paraffin lamps inside a greenhouse increased the rate of photosynthesis because of the release of carbon dioxide from the paraffin lamp. When we are burning something, carbon dioxide is produced, and therefore that carbon dioxide can be used by the plants for photosynthesis. Other areas in a greenhouse, they have some form of compression where they pump in carbon dioxide into the greenhouse. So thereby increasing the amount of carbon dioxide, and the increased carbon dioxide will have, and we'll see later how this affects us, will have a slightly higher rate of photosynthesis. Temperature, if there's a problem with temperature, it can also be increased by using heaters. And if temperature is too high, it can be decreased by using fans and or shade nets. So this is how the farmer can maximize using his or her knowledge on photosynthesis to get the best crop. This is what we mean by this heading. Let's go back to the heading. Maximizing growth. Remember, a higher photosynthesis will lead to higher growth, more crops, better for the economy, because then more can get sold and the economy improves in that way. And if we look at these factors that we are talking about, they are, notice from what we discussed here, they are carbon dioxide, light, and temperature. These are the limiting factors that we can manipulate in a greenhouse so that we can get things going in the process of photosynthesis. And I think with that, we can take a small break. Hi guys, and welcome back. I'm hoping you went to the toilet and you had a little coffee or you had a little tea, you had a quick chat, you phoned one of your friends, etc. Now, let's get serious again and let's get into this work. Just before we took the break, we were talking about how farmers can use the limiting factors of photosynthesis to maximize their yield, their crop yield. Now we're going to look at further into these limiting factors and how they actually affect photosynthesis. So the heading now here we're talking about are factors limiting photosynthesis. There are three factors that are limiting factors, and they are light intensity, carbon dioxide concentration, obviously, and temperature, as we spoke about just before we went to the break. Those are the three limiting factors. You may ask, yes, but what about water? Water is not necessarily a limiting factor. If there's no water, photosynthesis can't take place, end of story. They can't have too much water. They just have the water that they need for photosynthesis to take place and to keep the plant hydrated. If the water is going to affect the plant itself in bigger ways, so it's not a limiting factor as such. These three things, however, if they are manipulated, if they vary, then the rate of photosynthesis can be affected. Let's look at how, just with simple graphs. And again, guys, this is something you must take back home in a sense that a graph will tell you so much more than one paragraph can tell you. By just remembering these graphs, you will learn a whole lot about the limiting factors. Let's look at this, the first one, light intensity. So obviously this is amount of light intensity changing. This would be 0 and going up that way. And this is the rate of photosynthesis going up here. That's the x-axis and that is the y-axis. By the way, lots of people, when they think that you give a label for x and y, they simply put x and y and they think that's the label. No. When we talk about the labels for the x and y, we're talking about these labels here. Light intensity and rate of photosynthesis. In life science, generally, the physical factor will go on the x-axis. But that's just a trick to get away with it. What is it that actually goes on the x-axis? We want the independent variable. In any investigation, in any research, there are two variables that are studied. The independent variable and the dependent variable. So in life science, in the graphs, the dependent variable goes on the y-axis and the independent variable goes on the x-axis. Now, what is an independent variable and what is a dependent variable, you ask, obviously, because you're telling us this, but what are they? Independent variable is the variable that the researcher or the investigator or the scientist, whatever you want to call him, can manipulate, can change. In this experiment, we can change the light intensity, but we do not have control of the rate of photosynthesis. At the same time, to explain it in a different way, light intensity is not affected by the rate of photosynthesis. If the rate of photosynthesis changes, that does not affect, it won't change the light intensity. But if we alter the light intensity, if the light intensity changes, it affects photosynthesis. So the rate of photosynthesis is dependent on light, but light is independent of the rate of photosynthesis. This is how you need to understand that. Okay? So now let's look at this graph. If you can read a graph, you can draw a graph. And if you can draw a graph, you can read a graph. And in life science, the papers are generally loaded with graphs. So here we're saying light intensity, what's happening? This way it's increasing. Now what happens? What's the line here? As light intensity, if you have to explain any graph, the relationship between two variables, you say as the x-axis, light intensity, what's happening? Increases. So too does the rate of photosynthesis. Up to a point. Obviously, if there are figures given here, then you must use the figures in your answer. This one doesn't have figures. It's just telling you up to a certain point, after which it is constant. In other words, what we're saying, guys, that after this point, no matter how much more light, all this year, we increased the light here, all this year, it did not change that. Because remember, light is not the only limiting factors. Because we could be increasing light, but there's only so much carbon dioxide. So there's only so much this plant can do. So any more light is not going to make a difference. So we say that this part here, this point, is the optimum light. So once the farmer knows what the optimum light is, then he will focus on that light. It's no sense using energy to give more light than is required, because then you are wasting energy to make energy and to make food. So it's costing you actually more to get a crop. So you need to know what is the optimum and keep it at the optimum. Okay? Next, again, we don't have to explain independent, dependent. We've explained already. This one here on the x-axis, the level of carbon dioxide or the carbon dioxide concentration. And here, the rate of photosynthesis. What can we say here? Look at the graph shape. Not much. However, I'm going to change this graph slightly. There are certain cases where if we increase the graph that way, what will happen? It can actually come down. So what happens is, as we increase the rate of carbon dioxide or the amount of carbon dioxide, when CO2 increases, rate also increases. Then it levels. And any more increase can cause a decrease in photosynthesis rate from there down there. Now what happens here? If there's too much of carbon dioxide, the solution becomes acidic. Carbonic acid will form. And if we're doing that, then we are affecting the pH. And when we are affecting the pH, we will affect the enzyme reactions. So the enzymes will be denatured by higher or lower pH, in this case carbonic acid. The pH will be lowered, and therefore the rate of photosynthesis can eventually come down. So again, we need to know the optimum. We don't want to have too little, we don't want to have too much, we want just right. And the last one on temperature. Again, as temperature increases, the rate of photosynthesis also increases up to a point which we call the optimum point, after which any increase results in a decrease in the rate of photosynthesis. Why? We spoke about pH just now, and with pH came enzymes, and you know from your grade 10 work that temperature also affects enzymes. So as the temperature increases above, generally in most plants is about 25 degrees Celsius, is the optimum temperature there. After that, it will decrease because the enzymes become denatured. The enzymes become denatured. So these are the three graphs that we need to look at. Light intensity, carbon dioxide concentration, and the last one being temperature. So these are the limiting factors for photosynthesis. So what have we done so far? We've looked at word explanation, a simple explanation. We looked at the organ of photosynthesis, the leaf. We looked at the palisade mesophyll. We looked at the spongy mesophyll. We went inside the palisade mesophyll and spongy mesophyll, and we found this guy called the chloroplast. We went inside this chloroplast, and we found this, the outer membrane, the inner membrane, the stroma, the thylakoids, the grana. And we then found how that thing is suited for its function. From there, we moved inside the thylakoids and we look at how the light phase of photosynthesis takes place, how the light is used to make ATP and NADPH. From there, we went into the stroma, back into the stroma. There we saw how the dark reaction of photosynthesis takes place, using the NADPH, using the ATP, using carbon dioxide to make food. We looked at all of that. Then we looked at what are the limiting factors: carbon dioxide, temperature, and obviously the light that are limiting factors, and we saw how they affect photosynthesis. And then we look at how the farmers can use these three factors to maximize their crop. So that's what we looked at so far. Now let's look at the summary of photosynthesis then. This is the summary of the process, not what we have done. What I've just done now is the summary of what we've done. Now we're looking at the summary of this process. What happens? Light enters the chloroplast. The chlorophyll in the grana captures this light. And what does it do? Some of this energy is used to split hydrogen and, split water into hydrogen and oxygen. The oxygen is released into the environment. The hydrogen is carried to the stroma. Alternatively, the energy could also be carried into the stroma as ATP. That's the other cycle. One cycle, another cycle there. And here, the ATP and the hydrogen from here will combine with carbon dioxide to make carbohydrates. The carbohydrates are carried to the cells of the plants to do whatever plant needs to store it, whether it needs it to for respiration, etc. By the way, this carbon dioxide that we're talking comes into the leaf via the stoma. So in a nutshell, this is what happens during the light phase and the dark phase. There's the light phase there. That's all the light phase, and below that is the dark phase. Dark phase is taking place where? In the stroma. The light phase is taking place where? In the thylakoids. Not written here, but we say in the thylakoids. So again, a summary like this is useful for you to use to understand what is actually happening in this section. Now that we have done that, okay, and here's one more summary showing you the whole process in a diagram form. And again, guys, I keep saying this, that diagrams, graphics, speak much more than a page of notes and notes and notes. The diagram sticks in the head. The more color you add, the more this color may help you to remember certain things. And this one here is very easy. First of all, there's a light reaction. In a light reaction, obviously light is essential, and we're having the electron transport chain. One is where ATP is made, and the other one is where NADPH is made. In other words, one is non-cyclic, the one on the NADP, where water is split into hydrogen, the H there, and the oxygen there. The oxygen is released. This ATP and NADPH to leave the thylakoids and they go into the stroma. There you see them going into the stroma there. Also that comes into the stroma is carbon dioxide. This here is a scientific name for ribulose diphosphate. Again, I just said that to make you scared. Ribulose diphosphate, and to look smart. You don't have to know that word ribulose diphosphate. You simply say that the ATP and the NADPH combine with carbon dioxide in a series of reactions to give rise to sugar, glucose, which can become starch, which can become amino acids, which become protein, which becomes fatty acids, and they go to other cells in a plant where they would be used to do work. Now, why then, we asked the last question that we had in our bullets at the top was, why is photosynthesis important to organisms? Why is photosynthesis important to organisms? And the answer, first of all, most important, it maintains the oxygen and carbon dioxide balance in the atmosphere. Now you're asking yourself, what is he talking about? How the hell does photosynthesis maintain the oxygen and carbon dioxide balance in nature? Easy, guys. What does photosynthesis need? It needs carbon dioxide. And during respiration, what gas is given off? Carbon dioxide. Now in the atmosphere is all our gas of carbon dioxide, and the plants, when they have respiration, they also give off carbon dioxide. Now this carbon dioxide is in the atmosphere and it's in a high level. So if the plants photosynthesize, they use the carbon dioxide. So what do? They reduce the carbon dioxide levels in the atmosphere. At the same time, when they photosynthesizing, they release oxygen, which they themselves and you and I and other animals can use for respiration. Because for respiration, oxygen is used and carbon dioxide is released. And in photosynthesis, carbon dioxide is used and oxygen is released. Now you must understand this is a very, 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 very common misconception. Most learners, when they come to high school, they've learned in primary school, and they say it like this: plants take in carbon dioxide and release oxygen, and animals take in oxygen and release carbon dioxide, which is not totally incorrect, but it's not about plants and animals, it's about processes. The process photosynthesis uses carbon dioxide and releases oxygen. The process respiration uses oxygen and releases carbon dioxide. So whether it's a plant that's respiring or an animal that is respiring, in oxygen, out carbon dioxide. And obviously we can't see animals photosynthesize. They can't. Only plants, green plants, can photosynthesize. And when they photosynthesize, and it's not about them being plants, but it's about the process photosynthesis, which uses carbon dioxide and releases oxygen. So then that way, they maintain this balance between oxygen and carbon dioxide. That's why in the ecological world, in the world of agriculture, in the world of ecosystems, we talk about deforestation and how negative the impact is on the level of carbon dioxide. And there's too much carbon dioxide that leads to the greenhouse effect, etc., 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 which you will learn under ecology, not here. But can you see how our work is linked? That if it wasn't for photosynthesis, this balance, and that is why the more we chop the trees, less photosynthesis, more carbon dioxide in the environment. This is what we are talking about: global warming, the greenhouse effect, and all those things that go together there. And more important than even that, well, I can't say none of these are more important than the other one. They're all equally important. But the fact that the plant can use the sun's rays to manufacture food is what makes food available to you. Yes, you and I. It makes it available to you. You can't take your clothes outside and go in the sun and stand there and say, well, let me trap the radiant energy from the sun and let me absorb it and convert it to chemical potential energy. Only green plants can do that. And therefore, we all the heterotrophs, all those organisms that cannot manufacture their own food, they are dependent on the plant as a source of food. So all the food, we either get it directly if we eat the plant itself, or indirectly if we eat an animal that eats the plant. Okay? So when you're eating sheep and you're eating goat, when you're eating cow, the cow got its energy from where? From the plant. It's a herbivore, so it's eating plant. And when we eat the meat, that meat had its energy, the food, from the plant. And that's how it goes. Okay, so that's the first question. And it also deals with our theory. Now, the next question wants you to draw a drawing skill. Draw a label diagram to illustrate the general structure of chloroplast. And there we have a drawing. Remember, and I put this drawing here deliberately so that you know that we're not asking you to be an artist when you're drawing. And you and I can draw the same diagram. Let me show you quickly. One membrane. Remember, in life science, this part here is not allowed, but on the screen it's very difficult to get it right. But in your book, you can do that. You must make one line. Let's see if I get it right now. There we go. More or less, I got it right. So you're drawing your inner membrane and your outer membrane. You're going to label it. I'm not labeling it. Then you can draw lines. And you join the lines with lines. And then you're drawing your grana. There's your grana, and each line is a thylakoid. And you give it a heading, structure of chloroplast. Can you see that it's not a, you don't have to be an artist to draw this. I am by no means an artist. And I got I think, a beautiful diagram there of the chloroplast. And you can also draw the same. So that was that question. Good. This one here, this question also requires a little bit more in-depth reading. And these are the type of questions, each paper, paper 1, paper 2, will come with an example like this, where there's a lot of text that you have to read. So your language skills must brush up so that you can understand this. And the first and most important thing, like my good friend Kathy used to always say, that whatever comes part of the question is part of that question. Whether it's the text on the top, whether it's a diagram, whether it's a table, whether it's a graph, and the questions themselves, they all form part of the question. So you have to read it. Okay? So let's read this particular one. Okay, we got the pen on. A learner performed an investigation in which a green leafy twig from an aquatic plant was placed in a beaker containing sodium bicarbonate. The apparatus was exposed to sunlight, or light source, sorry, for a period of time, and the intensity of light from the light source was increased at regular intervals. What am I doing when I'm underlining all that? I'm identifying the key words in the question. Gas bubbles were released. The number of gas bubbles released per minute was observed and recorded. The results of the investigation are shown in a table below. Study the table and answer the questions that follow. So what did these guys do? They took a green leafy twig from an aquatic plant. They put this in a beaker containing sodium bicarbonate solution. This sodium bicarbonate solution is there to release CO2, to give more carbon dioxide. We'll talk about that. What did they do after it? What did they change? They changed only the intensity of light. So from time to time they put a stronger light, stronger light, stronger light. And they noticed that gas bubbles were released. They counted in a specific amount of time how much gas bubbles were released by each of that. And they recorded it. And there you have it, the light intensity, the different light intensities, 10, 20, 30, 40, 50, 60, 70. The number of gas bubbles released, 5 at 10, 12 at 20, 18 at 30, 24 at 40, 26 at 50, 30 at 60, and 30 again at 70. What is the aim of this experiment? Now, at this juncture, we need to explain the difference between an aim, an observation, and a conclusion. Many times when we ask for the aim in exams, we get the conclusion. When we ask for the conclusion, we get the aim. When we ask for the conclusion, we get the result. When we ask for the result, we get an aim or a conclusion. So we need to understand the aim. What is the aim of you going to school? What is the aim of you collecting money? The aim is, why are you doing it? So the question of aim, when you see the aim, you must ask yourself, why are we doing this experiment? What do we want to show? That's the first thing. Observation, what happens? What can we see? What can we smell? What can we hear? Result is the same thing. What happened in this experiment? That's the result. And lastly, the conclusion, because of your aim and what you saw, what can you say now? For example, let me give you a ridiculous example. I tell you, I aim to make R1 million in 5 years. That was my aim. Now, over the 5 years, somebody has to observe. And at the end of the 5 years, I need to make a conclusion. If you come to me and ask me now, you know your aim you gave me 5 years ago, what's your conclusion? If I tell you I bought a Porsche, is that a conclusion? No. It's not tied up to my aim. I can't tell you I went to town. It's got nothing to do with my aim. So your conclusion must be based on your aim and as a result of your results. So if I tell you at the end of 5 years, you know what, I only managed to make 500,000. It was, my aim was million. I look at my account, I see there's 500,000, and therefore I only managed to collect 500,000. That's my conclusion. So in the same way, you must think about that when you're answering these questions, okay? So what is the aim of this experiment? What were we checking? The number of bubbles that were released, and the number of bubbles indirectly tells us the rate of photosynthesis. So the aim of this experiment was to determine how light intensity affects the rate of photosynthesis. Notice, rate of photosynthesis matches with number of gases, bubbles released, and light intensity is given to you there. So we use the two variables in the experiment to give the aim. Name the gas that is released in the investigation. If you know your photosynthesis, there's only one gas that's released, and that is oxygen. What is the function of the sodium bicarbonate solution? To increase the amount of carbon dioxide. Explain the relationship between light intensity and the number of gas bubbles as shown. I love those questions. I told you, when you answer any question with relationship, you start with as. So what happens here? As the light intensity increased from 10 to 60, the number of gas bubbles also increased. Thereafter, it remained the same. So let's write that down. As light intensity increased from 10 to 60, the rate of gas bubbles released also increased. Thereafter, it remained the same. You notice, guys, I told you earlier when we were talking of the graphs on carbon dioxide, temperature, there were no figures given. Then you don't talk about figures. But in this particular table, there were specific figures given. So we are saying that as they increase from 10, increase to 20, increased, increased again, increased again, increased again, increased again, but here it remained the same. After 60, it remained the same, according to this table. So that's how you answer it. How would you set up a control for this investigation? Now, what the hell is a control? A control, first of all, is to have the same apparatus, but omitting the factor we're testing for. Now, in this particular experiment, how do we know that those gas bubbles come from the plant? Do we know this? We are assuming it because during photosynthesis, oxygen is released, and that is shown as gas bubbles. So the only way to know that that gas bubbles came from that particular plant, we can set up the same apparatus, but we don't put a plant inside. So in other words, we will put in a beaker, we'll have our sodium bicarbonate solution, and we will have no plant there, and we can check that. Alternatively, somebody else can say we can have the same apparatus with the plant inside, but we don't change the light intensity. We do not change the light intensity, we leave it the same. So we can see in a time period whether the gas bubbles are increasing or not. But I would prefer to go with the one where we are proving that gas bubbles are coming from the plant, because we don't know for sure whether it's coming from the plant or it's just gas bubbles coming. So the only way to know that is remove the plant. Good? Now we're going on to another investigation, and these questions here are testing your practical work in photosynthesis. Okay? So we're saying here, during an investigation, a pot plant was placed in a dark cupboard for 3 days. One of the green leaves from the pot plant was then partially covered with a tin foil. After being put in a dark cupboard, after the plant had been exposed to sunlight for 2 days, the leaf was removed from the plant and treated as shown in the diagram. So we take in the leaf, remember it had a tin foil there. We then put it in boiling water. We then took that out of there and we put something there. There's something A. The boiling water, by the way, is in a beaker, and there's another smaller beaker there in which the leaf is and substance A is inside there. And we took it out of there and we rinsed it out from there. We use a dropper with solution B and we put it on this leaf. Name the chemicals labeled A and B. I need to have that there. Good, so you can see both. A is a chemical inside this beaker. B is a chemical inside this dropper. And they are relatively, respectively, A is alcohol. We boil it in alcohol, but we do not expose it directly to the flame. And B is iodine solution to test for starch. Why is substance A used in this investigation? Why do we use alcohol? To remove chlorophyll. Oopsie. All right. Why is substance A used in this? Is to remove chlorophyll. Alcohol tends to remove the chlorophyll. By the way, if you do this experiment, you'll notice after you boil it in this solution that the alcohol becomes green. That shows that the chlorophyll has left the plant. And moving on, which part of the leaf, the covered part or the uncovered part, will give a positive result? In other words, they want to know which part of the leaf, all this here, or this area here. This is the covered part and that's the uncovered part. Remember, the covered part will not receive sunlight because it's covered with foil. So they want to know which part will give a positive result. And obviously the uncovered part because it's receiving sunlight. Name one precaution you would take when performing this investigation. First of all, you will not boil alcohol directly over the flame because alcohol is inflammable. It will burn, explode. Boil the test tube with alcohol in a water bath, any one of that same thing. Use forceps to remove the leaf from the hot alcohol and boiling water because you will burn otherwise. And also when you're using a Bunsen burner, you must be very careful that you do not get anybody else too close, etc. You must be careful. If you're using a Bunsen burner, first put the lighter on and then turn the gas on. Don't put the gas on first and then turn the lighter on because you'll have a whole ball of gas and you can have an explosion in that way. And whenever you're doing any activities like this, don't mess around in the lab. It's not a place to mess around because serious harm can be formed there that way. Secondly, or rather question 5, describe the process of photosynthesis from the time light is absorbed until carbohydrates are formed. Notice the mark allocation here, 18. No tabulation or flow charts will be accepted. So this question is obviously an essay question, and we need to learn the skill of answering an essay question. Notice what we've done. We have broken it up into the light phase and the dark phase. From the time light is absorbed, light phase, until the carbohydrates are formed, dark phase. So we start off by explaining the light phase. Where does it take place? In the grana or in the thylakoids. It's all given to you. Radiant energy is absorbed by the chlorophyll molecules and converted into chemical potential energy. The energy is used either to split water, photolysis, into hydrogen and oxygen, or to form ATP. This is also called photophosphorylation, another big word. Photo using light, phosphorylation to add phosphate to ADP to make ATP. Oxygen is released into the atmosphere, and the energy-rich hydrogen combines with coenzyme NADP to form NADPH. Then we go into explain the dark phase. It takes place in the stroma. Carbon dioxide from the atmosphere combines with the hydrogen from the light phase, using energy from the ATP came from the light phase to form carbohydrates such as glucose, fructose, sucrose, starch. All these reactions are controlled by enzymes. Notice 15 marks given for all your facts and 3 marks for synthesis. The synthesis is not a heading on its own. It's not something you have to write. The marker is given a rubric to judge your essay. Okay? First of all, he's going to look at whether you have given all the necessary information. Have you discussed the light phase and the dark phase? There were 2 things to discuss. Have you done that? Secondly, he's going to look at whether your information has gaps. Thirdly, he's going to look at whether your information has irrelevant. That means you started talking about the light phase and the dark phase, but in between you said stories about the leaf. The leaf is this and the leaf is that, and it has a chlorophyll, and you gave a whole lot of other things that are not necessary, irrelevant information. Also, fourthly, he looks at the logical sequence. Now, obviously, follow the question. The question says, from the time light is absorbed until the carbohydrates are formed. Look at our essay. It starts with the light phase, where light is absorbed. Then it moves to the dark phase, and look at our end. Carbohydrate, glucose, fructose, sucrose, starch is formed. So that is what we are talking about. It must have this flow. So when you look at the essay, look at the verbs. Whatever comes after the verb is what you have to answer, and follow the sequence in the essay so that you have a general flow. The sequence in the essay will give you your flow. Follow that flow. If there are 2 things to discuss, discuss both. If there are 3 things, all 3, etc., etc., etc. And make sure that you stick to your points given in that way. You can write brief sentences, but the sentences must make sense on their own. You can't just write one word there, stroma. What does that mean? It doesn't mean anything. The light phase takes place in the thylakoids. The dark phase takes place in the stroma. It must stand alone on its own. And guys, we move to the last question. And this question now, again, 2 potted destarched drenarium plants were well watered and placed on a brightly lit windowsill. A dish of sodium bicarbonate solution was placed on the soil, while a dish of soda lime, sodium bicarbonate, soda lime was placed on the soil besides the plant in apparatus B. Each plant was covered with a transparent plastic bag as shown in the diagram. There we have the diagram, sodium bicarbonate, soda lime. And in this experiment, they're telling you sodium bicarbonate gives off CO2. So it releases CO2 into this environment. Whereas soda lime, it absorbs CO2. So all the CO2 that's in this bag will go to the soda lime. So the question is, what is the aim of this investigation? To investigate whether carbon dioxide is essential for photosynthesis. Why were the plants left on a bright lit windowsill to enable the plant to obtain the light, which is a requirement for photosynthesis. After 48 hours, the leaf from each plant A and B were tested for the presence of starch. Right, that there describe the procedure to test for the presence of starch. We discussed it in a question before, and they are: boil the leaf in water, boil the leaf in alcohol, remember in a water bath, rinse in water, pour a few drops of iodine solution on the leaf. This is how we test for starch. This is the mother of all tests in photosynthesis. In which plant, A or B, would the leaves remain brown after the starch test? Obviously in B, because there was no carbon dioxide. We can say that B is the control in this experiment. Where there be a higher concentration of oxygen. In A, because photosynthesis is taking place and oxygen will be released. Give one reason for your answer to question D2. I gave it to you already. In apparatus A, photosynthesis occurs at a greater rate than in apparatus B, and more oxygen will be released. Okay, so that's the answer for the last one. Let's go back to the picture. In A, because photosynthesis is going to take place, more oxygen is going to be released here. While in B, there's no photosynthesis because the carbon dioxide is being removed, and therefore no photosynthesis will take place. Again, guys, experiment. This will be the experiment. This is the control. Why? Because we are investigating carbon dioxide. In this one, we allow carbon dioxide to be there, the experiment, and in this one we only change one thing. That means the plants must be the same, the pot must be the same size, they must be in the same area for light, but we remove the carbon dioxide. So when we take that one factor out, that is the control. I'm hoping that this session has given you more insight into the section, and I'm hoping that by way of the questions you have got more skills to equip you for this section on photosynthesis. With that, guys, have a great day.